Welcome back to the deep dive. Today, we are strapping ourselves into the world's tiniest and maybe most successful delivery trucks, mRNA lipid nanoparticles, or LNPs. We all know their recent smash hit. They delivered the COVID-19 vaccines with, incredible efficacy. But here's the massive problem. While these little delivery vehicles are absolute masters at protecting their fragile cargo, the mRNA, they are, frankly, terrible navigators.
And that navigational failure is really the scientific hurdle we're tackling today. When you administer these standard LNPs systemically, that means they go into the bloodstream, usually intravenously, they have this just overwhelming tendency to pile up in two main places.
Right.
The hepatocytes, which are your main liver cells, and then your antigen-presenting cells, which are, key for the immune response.
And this inherent liver bias, it just limits almost everything else we want to do with this technology. If we can't tell the LNP to bypass the liver and go specifically to, say, a cancerous tumor or a damaged pit of the heart, well, we can't unlock its potential for things like cancer or genetic disorders.
Or even critical cardiovascular issues, like delivering VEGF-A to heal damage after a heart attack.
Exactly.
So our mission for this deep dive is to map out exactly how researchers are solving this geographical problem. We're exploring the concept of rational control, so the established and the emerging techniques that let us dictate three things: where the LNP vehicle goes, where the mRNA message gets expressed, and even how the final protein behaves.
Okay, and just as a quick refresher, an RNA LNP is basically a nanocarrier, right? Super tiny.
Super tiny, yeah. Typically around 80 nanometers in diameter. And it's built to shield that really fragile mRNA cargo and, help it get inside a cell.
All right, so let's unpack this tiny delivery truck. If you were building the perfect biological nanocarrier to protect something valuable and negatively charged, what are the four essential parts you need? What's the blueprint?
It's a really ingenious combination of chemistry and physics. So LNPs are built from four main components. You can think of it like building a high-tech water balloon. You need the main wrapper, some structural stabilizers, something to give it rigidity, and critically, a stealth cloak.
Okay, so let's start with that main wrapper, the part that holds it all together, the ionizable lipids.
They're their absolute key component, making up about half the LNP by mass, and they do two really important jobs. Job one is cargo binding. These lipids are designed to be positively charged only when the pH is low, so below their pK. During manufacturing, we keep the pH low, so these positive lipids just snap onto the negatively charged RNA backbone, forming the core. And job two, which is so critical, is endosomal escape. We'll definitely come back to that.
So the ionizable lipid is the glue. What about the stabilizers? What helps it survive in the bloodstream?
For that, we use helper lipids, which impact the surface charge and add stability to the shell, and then cholesterol. Cholesterol makes up a big chunk of the LNP, and it affects the rigidity of the membrane. Yeah. You need it to be just stiff enough to stay intact.
And then there's the stealth cloak, the PEG lipids.
Exactly. The polyethylene glycol lipids. They enhance stability, but more importantly, they influence the protein corona.
The protein corona. That's the layer of proteins that immediately glom onto it in the blood, right?
Right. The second the LNP hits your bloodstream, it attracts proteins, and they form this coating. The PEG lipids, they kind of keep that corona from forming too quickly, which makes the LNP invisible for a little while. And that really helps determine where it goes and how long it circulates.
So what does this thing actually look like on the inside? Is it just a smooth bubble?
No, not at all. High-resolution imaging, like cryo-EM, tells us it's much more complex. There's an amorphous core of neutral lipids and cholesterol, and that's surrounded by the RNA, which is sandwiched between layers of those positively charged ionizable lipids, and then you have the outer shell with the other lipids and the PEG cloak.
Okay, so let's follow the journey. We've just injected this perfectly packaged LNP. What's its fate in the bloodstream?
The LNP immediately starts interacting with all those circulating proteins. That PEG stealth cloak starts to shed, we call it desorption.
Ah, okay.
And once that happens, protein binding just accelerates, and the clearance cells, so the liver's cleanup crew, your hepatocytes and specialized phagocytes like Kupffer cells, they start to internalize it.
Which brings us right to the biggest bottleneck in the whole process, the endosomal escape problem.
It's notorious. So the LNP is successfully absorbed by the cell, which is great. It lands inside this little protective bubble called an endosome. But once it's inside, up to 98% of the RNA LNPs fail to escape into the cytosol.
And the cytosol is where all the action happens.
It's where the machinery for translation, for actually making the protein lives. If the RNA doesn't escape that endosome, it just gets destroyed. It's useless.
So we're relying on a tiny 2% success rate. What dictates whether it's part of that successful 2%?
It comes back to those ionizable lipids. Inside the endosome, the pH naturally drops, which makes those lipids positive again. They then interact with the endosome's walls, disrupting and eventually rupturing them, allowing the RNA to spill out. So the specific chemical structure and the pKi of that lipid are what determines if that 2% escape is actually enough to be therapeutic.
Okay, let's do a quick recap. Before we get into how we can solve these problems, let's just nail down the key takeaways.
Sounds good. First, the structure. It's an 80-nanometer bubble. 4 key lipids protecting a negatively charged RNA core.
Second, the mechanism. Those ionizable lipids are the star player. They bind the cargo, and more importantly, they are what allows for that crucial endosomal escape inside the target cell.
And third, the massive limitation. Only about 2% of the RNA actually escapes that endosome, and the default destination for the whole vehicle is, overwhelmingly, the liver.
So if the liver is the default, how do scientists even know where these tiny delivery trucks are going in the first place? And, if they're even dropping off their payload, that tracking data must be crucial.
It's absolutely essential. And you have to distinguish between two different things: physical biodistribution and functional biodistribution.
Okay, what's the difference?
Physical distribution tells us where the LNP material is, where did the truck actually stop. But functional distribution tells us where the RNA is successfully being expressed and the protein is being produced. Where was the drop-off successful?
Wait, why would those two be different? Wouldn't the material just go where the protein is made?
Not always. And that's the key. The LNP can land in a cell, but if it fails that endosomal escape, the physical LNP material is there, but the functional protein is zero. So you have to track both.
So let's start with the physical material. How do you see an LNP deep inside the body?
For that, we rely heavily on nuclear imaging. It's non-invasive, it's quantitative, and it lets you see deep inside tissues. Researchers will radiolabel the LNP components, often with something like zirconium-89, and then use PET-CT scanners.
And what has that shown us?
Well, it really confirmed the LNP's role as a delivery vehicle. A classic study showed that when you injected bare mRNA, it just went straight to the kidneys for quick clearance. But when you put that same mRNA inside an LNP, it accumulated almost entirely in the liver. The LNP really steers the cargo.
That tells us where the truck ends up, but what about the timing, the kinetics at a cellular level?
Right. For that level of detail, you need something like electron microscopy. It gives you ultra-high resolution, but you need to use contrast agents, usually gold nanoparticles. And this gave us a really critical insight into liver uptake. It showed us that Kupffer cells, the liver's resident immune cells, showed really rapid, consistent uptake. They grab them right away. But the hepatocytes, the actual therapeutic target cells, they showed a much slower, continuous accumulation over several hours. So Kupffer cells are the fast responders. Hepatocytes are the slow and steady accumulators.
That timing difference is a huge piece of the puzzle.
Yeah.
Now, switching to functional distribution, proving the mRNA actually worked, that's where we see things like fluorescent reporter proteins, right?
Yep. mRNA encoding luciferase or GFP is sort of the workhorse, gives you a quick signal. We also use these elegant genetic switch models like the AI-14 mouse. It's a really powerful tool for preclinical research.
How does that work?
You deliver an enzyme that flips a permanent switch in the cell's DNA, causing it to glow, usually red, forever.
But the criticism there is that the permanent glow might, overstate the actual therapeutic effect.
Precisely. If a stem cell takes up the LNP and starts glowing, all of its daughter cells will also glow forever, even if the actual therapeutic protein, which might only last for 12 hours, is long gone. So it tells you which cell type got hit, but not necessarily for how long or how intense the effect was.
And for screening, when they're testing thousands of different LNP tweaks, how do they do that efficiently?
That's where barcoding comes in. It's really clever. They create a library of different LNP formulations, and each one is tagged with a unique DNA or RNA barcode.
Like a license plate.
Exactly like a license plate. They inject them all at once, and then they can go back later, take a tissue sample, and use deep sequencing to see which barcodes, which formulations landed where, and had the best functional expression. It speeds up discovery exponentially.
Okay, another quick recap. The key lessons on tracking seem to be:
1. Physical doesn't equal functional. Just because the LNP material is in a cell doesn't mean the drug is actually working.
And 2, timing matters. The immune Kupffer cells are the fast-acting cleanup crew, while the therapeutically relevant hepatocytes take a slower, steadier approach to uptake.
Right. So since we know the LNP's default road is this Kupffer hepatocyte highway, the big question is, how do scientists finally get behind the steering wheel and tell it to bypass the liver and go where we actually need it to go?
We're moving from just tracking the truck to actually giving it a destination address. And this seems to happen by changing the LNP shell itself, adding molecular address labels, or even using like external remote control.
Let's start with the chemistry. This is where tiny structural changes can have these huge geographical consequences. We've learned that just modifying the ionizable lipid can totally dictate where the LNP lands.
You mean like the chain length example?
Exactly. Researchers found that if they use multi-tailed ionizable phospholipids with shorter alkyl chains, the LNPs mostly go to the liver. But if they switch to longer alkyl chains, expression shifts significantly to the spleen.
Oh.
It's a massive change from just making the lipid tail a bit longer.
And this is where it gets really fascinating for me, the bond type switch.
It's absolutely incredible. By swapping a common ester bond for an amide bond in the lipid, they created what's called an N-series lipid. And that lipid targets the lungs. The original O-series with the ester bond targets the liver.
And why? What's the mechanism?
It all comes back to that protein corona. The different chemistry creates a unique protein coding in the bloodstream. And that protein coding basically acts like a new ID badge, telling different cell receptors whether to recognize and internalize the LNP.
So that's the passive chemical steering. What about active targeting, giving it an explicit address label?
For active targeting, you decorate the LNP surface with molecular addresses, things like antibodies, peptides, or aptamers. A really great example is targeting hematopoietic stem cells, or HSCs, which live in the bone marrow.
So how did they manage to get past the liver and into the bone marrow?
They decorated the LNPs with anti-CD117 antibodies, and when they compared it to a control LNP, which, predictably went to the spleen and liver,
Right.
the targeted anti-CD117 LNPs showed this dramatic increase in expression in the bone marrow, but also the lungs, intestines, and skin. It was a true rerouting success.
But I imagine adding these complex addresses must have some trade-offs.
Oh, for sure. The main one is stability and cost. Finding the right ligand is expensive, and sometimes adding it can make the LNP less stable. Plus, that protein corona we keep mentioning can actually cover up the targeting ligand, making the address label invisible to the target cell.
Okay, so finally, let's talk about crowd control. How do you direct the flow of traffic away from the liver?
You can use inhibition or priming. A great example is nanopriming. You give a dose of unloaded liposomes about an hour before the real LNP dose. These decoys go in and they transiently occupy the clearance cells, the Kupffer cells. It's like blocking off the parking lot.
I see.
And this reduces the liver uptake of the actual drug, letting it circulate longer and improving delivery to other organs. We can also use things like Socus ultrasound to temporarily open up cell membranes, essentially acting like a local spotlight to force delivery only in that one specific area.
So we've mastered steering the vehicle. Now we move to writing the actual message. It sounds like synthetic biology is providing this ultimate safeguard, making sure that even if the LNP lands in the wrong place, the message isn't read, which prevents off-target toxicity.
That's exactly it. The solution is to write instructions directly into the mRNA itself, making translation strictly cell-dependent. And the most widely used technique here involves microRNA target sites, or mRTs.
How do these mRTs work as a stop sign?
So microRNAs, or mRNAs, are small natural sequences that cells use to suppress specific mRNA translation. If we engineer sequences that are complementary to a specific mRNA into our therapeutic mRNA, any cell that expresses that mRNA will recognize our sequence and immediately silence our synthetic mRNA. We're effectively detargeting it.
Can you give us a really powerful example of that safety mechanism in action?
The HSC targeting study is the perfect one. The original goal was to deliver PMA-RNA, a gene that makes cells self-destruct to deplete stem cells. But even with good targeting, the little bit of residual liver uptake was lethally toxic. The fix. They added a target site for miR-122, which is highly specific to liver cells. This completely silenced the PMA in the liver, preventing the toxicity, and it enabled a selective, non-toxic depletion of the stem cells in the bone marrow. It was beautiful.
It's like we've moved from just steering the truck to writing a full biological operating system for the mRNA. So what about these sophisticated ADAR sensors?
The ADAR sensors are a fantastic example of a biological on-off switch that uses the cell's own editing machinery. Researchers designed the mRNA with a premature stop signal. So by default, this message will never be read. But they also build in an engager element that's complementary to a trigger RNA that only exists in the target cell.
And what happens when those two RNAs meet?
When they hybridize inside the cell, a little mismatch forms right at that stop signal. The cell's natural editing enzyme, called ADAR, sees this engineered mismatch and it corrects it, which removes the stop codon. and suddenly translation is turned on.
Incredible. So it only works if that specific trigger RNA is present.
Exactly. It offers this incredible cell-specific control.
So we can control where the vehicle goes, and we can control where the message is translated. What's the final layer of control once the protein has already been made?
This final layer is all about optimizing the protein itself for stability, location, and function after it's been translated.
Okay, so if the therapeutic protein is a gene editor like Cas9, it has to get into the nucleus to work. How do you ensure that happens?
We use something called subcellular localization signals. For the nucleus, we engineer a nuclear localization signal, or NLS, right into the protein's code. It's a short peptide sequence.
And it's like a VIP pass.
Think of it exactly like a VIP pass that lets the protein be actively ferried across the nuclear core complex. And the number and location of these NLS copies can strongly influence how effective the gene editing actually is.
What if the protein is highly toxic, like a cytokine for cancer therapy? You can't have that spilling out into the bloodstream.
That's where we use membrane targeting to limit what we call spillout. For these really potent cytotoxic cytokines like IL-12 or IL-15, we fuse them with a transmembrane domain. This literally tethers the cytokine to the surface of the cell that made it. It prevents systemic toxicity, and it concentrates the therapeutic effect locally, which dramatically improves the therapeutic index.
And finally, what about an emergency brake? Is there a way to build a kill switch into the protein itself?
Absolutely. For that temporal control, we use degrons. Researchers engineer the protein with a specific structural motif, the degron. Now, this degron is totally stable until you administer a specific small molecule drug, like lenalidomide.
And the drug activates the kill switch.
The drug acts as a molecular bridge. It forces the degron to bind to the cell's natural recycling machinery, and this immediately triggers the destruction of the protein. It gives you a reversible, drug-controlled on-off switch, and that's vital for highly toxic payloads where you have to strictly manage how long they're active.
Wow. We have truly charted the transition from a simple, if highly successful delivery truck to this incredibly sophisticated, multi-layered system that's capable of unprecedented control. We've seen that high-precision delivery involves tuning the LNP shell with chemistry and ligands, programming the mRNA message with MIRS and ADAR sensors, and finally optimizing the protein payload itself with localization signals and temporal control.
And this convergence of chemistry and synthetic biology is only getting faster. I mean, the sheer volume of data being generated from screening thousands of lipids to functional tracking, it's going to require AI and machine learning to interpret. The ultimate goal is to move from trial and error to having whole libraries of LNP and RNA formulations with specific, well-defined distributions to maximize that therapeutic index, high effect, low toxicity.
The journey from defaulting to the liver to precision molecular software is really changing how we view medicine.
It is. But we have to remember that for toxic payloads, like cancer therapies or permanent gene editors, the specificity we need is far, far more stringent than for a safe vaccine. We need near-perfect targeting and near-perfect silencing of any off-target effects.
So let's leave you with this final thought. We've seen that small chemical changes to an LNP, like swapping an ester bond for an allylamide bond, can radically change where it goes in the body. And we've learned that scientists can design mRNA that only translates if a specific trigger RNA is present in a specific cell. Considering all these layers of control, what is the least invasive, high-specificity application of this RNA-LNP technology, maybe treating a neurological disorder or regenerating a specific tissue, that you are most excited to see translated to the clinic?